pleasure to introduce Akanksha Jain. Akansha did her bachelor's at University of Delhi, or Delhi University, I don't know how is it exactly pronounced, but studying botany. And then after that, she came to Tata Institute of Fundamental Research in Bombay, where she studied motor proteins in Drosophila with Krishanu Ray. That's also where I met her, Akansha, for the first time. After that, she went to Max Planck Institute for Cell Biology and Genetics in Dresden, where she worked with Pavel Tomanchek, whom you might know from OpenSPIM efforts all across the world. Right? So she's has plenty of experience with different model organisms, but interestingly, now she's moved on to organoids, and she works with Barbara Treuklin at the University of ETH-Zueich, and still continues to use a lot of light sheet technologies to study developmental processes in organoids. So I wouldn't take much of your time. Just as a rule, we'll have the question and answers at the end. You can type in your name, and I'll point you and unmute you, or you can put your questions in the question and answer section, and I'll ask them for you at the end. Right? Okay, Akansha, take it away.
Okay. Hello, everyone. Thank you, Senthil, for inviting me for this seminar. I'm very happy to talk about and discuss the work I did on studying the red flower beetle in Pavel Tomanchek's lab. And also I will introduce a bit of what we are doing with organoids, imaging and analysis more recently during my postdoc. So a very fundamental question in biology, and something that absolutely fascinates me, is how diversity is created in life forms. So one can think of tissues very similar to the art of origami. So depending on how you fold a tissue or how you create folds in a paper, you can create many different types of 3D outcomes and 3D shapes. Now, this diversity is not only seen in adult organisms, but often embryos also mimic this tissue origami. So between different embryos, if you start comparing, then there are many different kind of folds and 3D shapes that are being formed and patterned to give the final shape and function of a tissue. So at the core of this morphogenesis, or the mechanism of creating shape, are cell behaviors. So what happens during development is that cells undergo different kind of behaviors, such as a cube cell could become a squamous or a columnar cell. And this results in the overall topology change of a tissue. So for example, an epithelial sheet can go from a cuboidal sheet to a more thin and squamous sheet, which is bigger in size. What can also happen is that cells change their shape to exchange their neighbors. This is called as cell intercalations, where two cells which were not originally neighbors come in touch. And this leads to a process called as convergent extension, where a tissue extends in one direction at the expense of the orthogonal direction. So these cell behaviors, or these cell shape changes, are brought about by the activity of generic effectors. So these are force-producing molecules in a cell, such as the actomyosin cell cortex, or adhesion molecules or integrins, for example. However, the effector molecules are very generic and they're present in all cells. So their specific spatial activity is controlled by regulatory genes. Regulators are patterning genes, which divide an embryo or a tissue into distinct genetic domains. And now in each of these domains, the activity of the effectors is controlled to bring about cell shape changes. So eventually what happens is, by a coordination of cell behaviors, effectors, patterning genes, and the mechanical properties of a tissue, many different 3D shapes are created. And these distinctions are not only between organisms, but in the same tissue over time as an embryo or an organism develops. And also, as I will show in this talk, these distinctions are also often present in the same tissue in different regions. So to study this, we started by looking at the embryogenesis in the red flower beetle Tribolium. Now, why Tribolium? So Drosophila is a very well studied insect system, and the embryogenesis is quite well understood. But actually, Drosophila is very atypical, and it is not a very good representative of how most insects develop. Rather, Tribolium is a better representative, and it also undergoes a dramatic tissue epiboly, which is not seen in Drosophila. So here you see a Tribolium embryo with cells labeled for the membrane. Now the anterior part of this embryo, denoted by this line or this cap, is this anterior roughly 1/3 cap. As the embryo develops, this cap forms an extra embryonic tissue. So this tissue is degraded at the end of embryogenesis, as is not present in the larva. This tissue cap then expands in all dimensions to form an outer shell, only this bottom layer, the bottom thin layer, is actually the embryo undergoing gastrulation. So this layer is engulfed inside, covered by the protective shell, and inside the embryo undergoes gastrulation and germ band extension. So this process is very much unlike Drosophila, but it's actually very similar to vertebrate systems and zebrafish or other amphibian systems. So in zebrafish also, the EVL layer expands and covers the embryo. What is known from studies in zebrafish is that as this EVL layer expands, a lot of mechanical stress is created in the tissue. But this stress is released by cell divisions, which are oriented along the direction of the stress. But we know from studies in Tribolium that Serosa expands, this extraembryonic cap expands only through cell flattening. So no cell divisions are seen in Tribolium, although a massive cell sheet is expanding here as well. So how is this mechanical problem solved? And not only a mechanical problem, there is also a very big geometric constraint here. So now think of this tissue. So this in this cartoon, the serosa tissue is the green tissue. So as this tissue starts expanding or goes from the pole to the equator, the tissue has to start solving two problems. The dorsal side of the tissue closer to this arrow is consistently expanding, as you can also see in the cartoon here with the blue region. But if you look at the leading edge of the tissue, or the edge which will undergo closure, this edge has to start contracting. So while the same tissue is expanding in most regions as it covers the embryo, closer to the window, this tissue now has to start shortening in size and close this window to create a seamless epithelium or a very seamless outer shell. So as you can already appreciate here, this problem is a very complicated 3D problem because the serosa is expanding dorsally, laterally, and closing on the ventral side. So all directions of the embryo have to be covered. So how do we study this and how do we image this? So of course, what we did, we went to light sheet microscopy, and I have mostly used for all of this work the Zeiss LZ1. So what we did here is you suspend an embryo or you mount the embryo in a capillary in agar, and then you push it out and you turn it around on its long axis. So with the objective, you can collect multiple angles of the same embryo. We then take these images and process these images using Fiji. So this is all open source plugins that are available through Fiji, and you can very easily use them to stitch your data. What we do for the perfect alignment is to add beads to the agar medium, which you can see as these fluorescent dots outside the embryo. So here, for example, I collect 5 different angles of the embryo, and I use these geometric points provided by the beads to stitch them into the final 3D embryo. So once we do this kind of processing, you can end up completely covering the dorsal, ventral, or all sides of the embryo, and not only the long or the dorsal ventral side, but the very difficult to image regions, which are the poles of the embryo. So after stitching, I can also look at the anterior and the posterior pole very nicely. And not only for a single time point, we are of course doing this for the entire duration of the Serosa expansion and closure, which amounts to roughly 16 hours of the beetle development. So all of this data then ends up being really huge. So most of these movies were above a terabyte that we wanted to then analyze and quantify. So on top of being heavy, another challenge with this kind of data is that with conventional visualization, you only look at a part of the embryo. So although this movie that I show here is a 3D movie, but I'm just looking at a single lateral view of the embryo. And this doesn't solve the problem because I want to look at that tissue in one plane and compare different regions of the tissue. So to solve that, what we did was to adopt cartographic projections, which is very similar to making maps of Earth. So what we do here is to fit a cylinder to the embryo and unroll that cylinder. Then not only we unroll one layer, but in fact, we peel it like an onion. So we go through the depth of the embryo and keep on peeling successive onion layers to create a flat 2D map, which also has the volume reconstructed. This makes now the tissue visualization really easy, and one can also color code it for different depth. So here in the beginning, the tissue is in one plane, so you see it as more yellowish. But as it develops, the top part of the tissue is now green with the outer serosa, whereas the embryo, which invaginates inside, is magenta. So this helps in visualizing just the serosa behaviors for the outermost shell. But we do not want to just visualize the maps, right? We don't want to just look at the data. We also want to quantify different behaviors across this tissue. But as we all know, maps are often distorted and we cannot make direct calculations on that. So to solve this, what we developed was a series of plugins in Pavel's lab, where we first, you can take an image or you can take a map segmented. So you can have the cell membranes or the nuclei segmented, and then you also export the distortion charts. So what's a distortion chart? So in the map projection, every pixel is of a different size because of unwrapping the curved tissue. But you can export this as the math as an image and combine it together to correct for the distortion and give the correct measurement to the value of a cell area or any quantity. So we do this, and now by drawing onto the map, we can figure out how big is the tissue area. We can also find out quantities like what are the cell area distributions, shape index and tissue fluidity, which I will talk about, as well as circularities and neighbor distances. We can also map back these projections to the 3D curved tissue again to visualize in the embryo how the distribution of the quantity is in different parts. So once we developed all of this system, we started looking at the serosa morphogenesis over time. So the first thing we quantified was to see how much is the serosa expansion. So we know this starts as an anterior cap, but what is the percentage of this cap and how much is it really increasing? We found that the serosa cap is actually roughly 1/3 of the total embryo area in the beginning, which means it goes under a triple expansion by the end of the window closure. However, this expansion in size by 3 times is not through cell divisions, only through the cube cells becoming flat and squamous and increasing their area. So then next we look at the cell area distributions across the tissue. So as you see here, the red box denotes the dorsal part of the embryo, which you also see in the cartoon at the top green region. So you see here, as the yellow intensity increases, that area on the dorsal side is of course increasing over time. We quantify this by going to the confocal and measuring a lot of cells in this region. But if you compare in the same tissue 2 different regions, so the dorsal to the ventral, which is closer to the window, then cells which are close to this window are actually much smaller in size. So this creates an interesting question, that cells which are closer to the window are smaller. So does this mean that as the window shrinks, the cells are just continuously becoming smaller to help in the closure? Or could it be that the cells are also decreasing in numbers? To check for that, we went back to the maps and we tracked cells across this boundary. So as this boundary goes from the dorsal to the ventral sides, we look at the cells and we see that by the end of the window closure, there are maybe just 5 to 7 cells left, which all came from very different and sparse regions on the boundary. So we quantified this and we found that in the beginning, the total number of cells forming this boundary is roughly around 75 to 80. But by the end of it, it is only 5 cells or so that are present as the closure has happened. This happens because cells which are close to the window are actually constantly exchanging their neighbors. So I have shown here in zoom in of the red box on the movie, where you see that the yellow and the blue cell actually had a cell in between them. But as the window closure proceeds, these cells exchange neighbor, so that by the end, the yellow and the blue cell are touching, whereas the in-between cell has moved out. What this means is that cells are intercalating and exchanging neighbors in the serosa in the same plane as the window. So we tracked this again on the maps, and now we are tracking a lot of cells close to the window in the leading edge, but also far away on the dorsal side. And we see that as development proceeds, as the window is closed, cells closer to the window constantly exchange their neighbors and get mixed up, whereas cells which are on the dorsal side remain rigid and maintain their neighbor contacts. So this is very reminiscent of behavior of molecules in a fluid. So you can think of the ventral region as a more fluid-like region, which is remodeling more and exchanging neighbors to adapt to the topology. Whereas the dorsal part of the embryo is more jammed or more rigid or more solid-like. So it doesn't exchange its neighbor, it maintains its contacts and just stretches over time. So to test for this, we adapted a theory which is based on work from Lisa Manning's lab, where they have postulated different parameters and theories for confluent tissues. So in this particular case, we measured shape index, which is the perimeter to the square root of area of a cell. So basically what this means is that if your shape index is higher than 3.81, then you are likely to be a fluid, whereas if it's lower, then you're more solid. When we check this for the serosa, we see that on the dorsal side, where cells don't exchange their neighbors, the shape index is lower and stays close to 3.81. Whereas on the ventral region, which is this leading edge, which is closing the window, the shape index is much higher. So indeed, in fact, it looks like that the region which is closer to the window could be behaving more like a fluid. So what does this behavior have an impact on the mechanical properties of the tissue, or to correlate how this relates to the mechanical properties of the tissue? What we did was to make large scale ablations across the tissue in the serosa as it develops. So here you see the dorsal side of the embryo. So the top part where the red box is. And over time, what we saw, so we cut this tissue and we measure the instant recoil. And this instant recoil serves as a proxy for the tension in the tissue. So we saw that as the serosa goes over the pole, the tension in the tissue is increasing, which means that over time, it becomes harder to pull this tissue and close it. But if we look at the ventral side, then on the same stage, compared to the dorsal, the ventral region has much lower tension. So the tissue fluidization and the rearrangements that are happening could be resulting in a lower tension on the ventral side of the embryo. So then we included this behavior of a gradation of tension to the theory, and we measured a parameter called as tissue fluidity. So by measuring tissue fluidity for each cell, we found that a closed band of cells, which is the leading edge, very close to the window, this edge is showing a fluid behavior, whereas the rest of the tissue stays more as a solid. So the obvious question for us was, what could be creating this very specific behavior close to the window? So we went back and we started looking at actin in these map projections. What we see is that between these two tissues, the embryonic and the extraembryonic tissue, there is a very supracellular cable-like actin intensity enrichment. So if we track this, we see that as the tissue develops over time, first, this boundary moves over the pole. So the actin cable increases in size. But as the window is closing, this cable starts coming together. It changes shapes quite a bit, becomes smooth, becomes round, and ultimately closes at the point where the window closes. We also went back and we asked, is this cable now a contractile cable? Does this cable have tension? Could it be pulling the tissue? So to test for that, we ablated the cable again and we measured its recoil velocities. What we see is that indeed it's a supracellular contractile cable, because over time, the tension in this cable is really high. So as the cable moves over and forms a circle to start closing, it really becomes much higher in tension compared to the earlier flat stages. Lastly, we looked at myosin in the beetle to see if this actomyosin cable could be having some special behaviors. And we found that the myosin intensity across the cable is very heterogeneous. So there are some cells which have higher myosin, which you also see in this chymograph. And what happens is that throughout this boundary, all the cells which have a really high amount of myosin, they contract faster. So they become a point and they exit the cable to remain in the plane, but start exchanging neighbors. So this means that all the cells don't shrink at the same time, but there is more like a sequential shrinking in different regions, leading to this sparse random exodus of cells, which promotes the intercalations. So with this model, we thought that it is the actomyosin cable, which is actually promoting the structural transition, the solid to fluid transition in the Tribolium serosa. So we looked at the literature and we found that there is a gene called as Tribolium Zen1. Zen1 specifies the extraembryonic tissue in Drosophila as well as Tribolium. So if you knock down Zen1 by injecting a dsRNA in the beetle, then you don't have the green tissue cap anymore, and all of the tissue is now the embryo. So once we did this, we see that indeed we do not see any enrichment anymore, and the window in fact remains completely open and fails to close. So then we measured the quantities that we showed for wild type before, and we see that all the cells closer to the window are now much smaller. They do not reorganize. Their tissue fluidity value is much lower. So basically what ends up happening is that the cable is breaking the tissue into two regions and promoting rearrangements. And these rearrangements are now missing because this actin enrichment, this contractile cable is missing. So in summary, what we have shown is that as the Tribolium embryo develops, the extraembryonic tissue expands over the pole and covers the embryo. Now, this tissue then shows two kinds of behaviors. On the dorsal side, the tissue behaves more like a solid-like contractile tissue, whereas on the ventral side, the tissue is more fluid-like with lower tensions and is able to remodel and close the window. This behavior on the ventral is promoted by the activity of a contractile actomyosin cable that leads to sequential eviction of cells and rearrangements. So here I would end the first part about the Tribolium embryo, and I will switch gears to talk very briefly about what I'm doing with organoids in Barbara's lab. So far what I discussed was very similar to studying a well-functioning machinery. So I have looked at different embryos and we have studied different developmental systems. And what we do is we mutate genes and we try to figure out what the function of that gene could be, or we try to get rid of a tissue and we see how the machinery breaks down. An alternative way to understand morphogenesis would be to start looking at synthetic tissue systems and build the tissues. So you have all the different cell types that are forming a tissue in a native scenario, but you do it in a synthetic petri dish and you try to create the tissue system. This has been now enabled by the recent developments and advancements in organoid systems. So more specifically, it has made possible the access to human development, because we are able to now generate different kinds of human organoids, such as brain or pancreas or retina, in a dish in the lab, and then control it. So what we are focusing on are human brain organoids. So studies from different labs have shown that if you aggregate cells in a spheroid, and then you induce it to a neural fate, you form a neuroectoderm, and then you just let it grow. You do not give it any patterning cues. The neuroepithelium is able to self-organize. So in contrast to an embryo, which has a lot of maternal positional informations, the organoids, the human brain organoids, are a completely self-patterned system, which is able to make lumen and organize cells in this lumen, very reminiscent of the human primary tissue and the human neocortex, as you can see here. Work done from Barbara's lab using stainings as well as single cell transcriptomics has shown that over a period of 2 months or 2 to 4 months, starting from pluripotent stem cells, the organoids form neuroectoderm and then diverge into different lineages. So after about a month, there are the early neuroprecursor cells, and these then start committing to the dorsal and the ventral forebrain, as well as the midbrain, hindbrain lineages and the retina lineages. What is unclear is how these commitments happen. So what happens in the initial 2 to 3 weeks of patterning that leads to the neuroepithelium committing to different brain regions, and how does this lineage commitment as well as the spatial organization happen? To study that, I started adapting the live imaging tools that we had established to look at the beetle embryos. Example here is a multi-view image of a human cerebral organoid, which I have collected again with the Zeiss microscope and stitched the data to look at the complete 3D overview. So in cross sections of the 14-day organoid, you can already start seeing the tiny lumen, which will later on expand and pattern to form the layers. We can also again make maps out of these organoids to study the topology of the different lumen as it develops over time. So the image here shows a depth projection, wherein magenta is the deeper part of the organoid, whereas the yellow is the topmost part of the lumen. Lastly, we are also using fluorescent cell lines and fluorescent iPSCs to start looking at nuclei and actomyosin behaviors as these organoids pattern. To do this live imaging, I needed to establish a system which would be stable from the time that you have a neuroectoderm, which would be a day 6 or a day 10 sphere, to roughly 3 weeks when it starts committing to different brain regions. So to do that, we have adapted an inverted light sheet system from Viventis Microscopy. And in this system, we are imaging organoids starting with the neuroectoderm for roughly 3 weeks. So the movie I show here is one such movie, which is pseudo-colored. So the green here are nuclei, and the pseudo-colored cells are the membranes. And as this organoid develops, so you see a cross section on the left side and a maximum projection on the right side. You see that different lumens start forming, and over time, this lumen also start thickening. So first, the neuroectoderm proliferates and it becomes bigger, and then these cells start becoming longer to start doing interkinetic nuclear migrations and start forming a pseudo-stratified neuroepithelia. So we next started analyzing this data, and what we did was, in combination with the genetic barcoding, So in the lab, there was developed a system called as iTracer, which barcodes individual lineages that you can track over time with single cell transcriptomics. Using this data, we figured out that in the organoid, lineages tend to commit very early on to a specific fate, and then they locally proliferate. To corroborate that, we went to the live imaging movie that I just showed, and we started tracking lineages with a framework called as Mastodon, which is again available in Fiji and was developed in Pavel Tomacek's lab at the MPI. So using Mastodon, we first tracked one lineage in one lumen, so the pink lineage here, or L1, for a period of 100 hours. And we saw that all the progeny of this lineage tend to remain restricted to the same lumen. We then tracked more lineages and also one nuclei in a diametrically opposite lumen, the green nuclei, and we saw that these lineages never tend to mix. So very similar to what we were seeing with the RNA-seq data, we actually saw that also with live imaging, you can recapture behavior in the organoid where you see lineage commitment happening pretty early on and only local proliferation and local enrichments. What is also very interesting here is that this behavior is actually very similar to what would happen in vivo in an embryo. So with this kind of approach, we finally start seeing that organoids during their development are also probably mimicking how embryos develop or how a neurectoderm would develop to form a brain. If you're interested more in this, I would be happy to discuss. We have this preprint now on bioRxiv, which shows more about the iTracer system, but also about the imaging that I discussed here. Okay. So in total, at the end, I would like to thank many people whose work I have shown here today. The Tribolium work was done with Pavel Tomacek. So I thank Pavel and Tassos, who were both my PhD supervisors, and the entire Tomacek lab. Especially, I would like to thank the Tribolium community, which is a very open community and shares reagents and knowledge, as well as the light microscopy facility at the MPI, which probably is incomparable in how it helps and how it helps a project grow. The organoid work is from my postdoc in Barbara Treutlin's lab and in collaboration with the CAMP lab in Basel. And here I would especially like to thank Viventis for helping us set up the microscope, which provides a very stable cell culture system for doing this kind of long-term imaging and analysis work. Okay. I am open for questions.
Yes, thank you, Akansha. It was a fantastic talk. Also, thank you very much. I know it's 2 a.m. over your place. Thank you very much for staying up tonight and giving this presentation. I'll just see the list and unmute people.
Yes.
And let them ask questions. The first question is from Alberto Rosella Diaz. Alberto, do you want to ask yourself? Yeah.
Yes, can you hear me?
Yes.
Yes.
Yeah, it was a fascinating talk. Thank you very much. I think there was a tour de force, pun intended. I guess the question in the first part would be, what triggers the appearance of the cable? And do you know if buildup of tension is required for the cable to appear, or is it totally genetic or autonomous?
Yes. So I think the cable appears at a tissue boundary. So what we know from other systems, mainly Drosophila, is that when there are heterotypic cadherin interactions, that leads to an actomyosin enrichment at tissue boundaries. So we have not studied this in the beetle, and primarily the reason is that we really lack reagents. So for this project, I cloned myosin for the first time in the beetle, and a lot of it was just visualizing myosin.
Sure.
But if I were to make a guess, I think the cable is really being enriched because of the genetic boundary between the embryo and the extraembryonic tissue. And that's why when we abolish the tissue, there is no cable.
Okay, cool. Thank you.
Next question is from Alex Coombs.
Thanks very much. Amazing talk. I just had some practical questions around tissue labeling and cell tracking across the two kind of model systems that you're working on. So the actin stains were just beautiful in the Tribolium. Was that something that was genetically encoded, or was it in the media?
Both, actually. So not in the media, because insect embryos have this vitelline membrane, which is like a waxy coating on the outside. It's transparent, but not much can go through that, unlike fish or other systems. So we did two things. The beauty of Tribolium is that you can make a plasmid, and you can synthesize RNA and inject the RNA. So a lot of the work I show is just by injecting the mRNA for myosin or live fat. But we also have a genetic line for live fat, which is the constitutively expressing under tubulin promoter.
Okay, and the follow-up question for that in the organoid context was whether you had tried, LifeAct or media-based actin stains, and if so, how do they compare?
Yes, so for the organoids, actually, we are using stable transgenic lines. So these are all, we have quite a few lines which label endogenous actin. So these are CRISPR endogenous tagging lines for nuclei, histone, myosin, or actin. I have not tried the media labeling method, mainly because, so I tried it a little bit for the beetle. And from my discussions with people and my initial trials, I know that very often it can be quite toxic. And toxicity during this imaging is very hard to estimate. So very often we do not know whether the phenotype we are seeing is really the phenotype or something due to the label. So I was a bit lucky that I could access these lines, and I am rather investing on creating reagents which would be endogenous tagged and not do the media approach.
Yeah, that makes sense. I'm happy to shelve my second question for a bit, or just ask it now. What do you?
Go ahead.
Okay. Yeah, so the second question was around cell tracking in the two different contexts. So with the Tribolium work, it just looked like you had a very, clean resolution of individual nuclei, and the cells were kind of moving in sheets. So it seems to be like it's perhaps a bit easier to image and probably easier to track. Is that true compared to the organoid work, where it was evident that you had, a bit of loss of resolution at those cells in the middle? And how do you actually approach that? Did you have to manually track your cells, or is it a machine learning type approach?
I have tried quite a few different approaches over the years. For the Tribolium part, we are pretty much restricting to the outer shell of the embryo. So it does make it quite easy, but the problem is at the window. So at the window, it's a curved sheet, and cells are often going in and out over time. So there it was quite hard, which is why I took this approach to differentially label the layers in colors. So I can see when a nuclei goes from the top layer to the inner layer. And then we decided to track on the flat map precisely to avoid the problems in the 3D tracking. Simultaneously, in Pavel's lab, this Mastodon was being developed. This uses TrackMate and Big Data Viewer. So a big issue is with the size of data. So often I am having something to deal anywhere between 1 terabyte to 3 terabytes, and then multi-channel or single channel. And this, for me, has failed with most commercial systems or other available solutions. What works very well is the Big Data Viewer in Fiji, which loads data in a pyramidal format. So Pavel's lab has built a lot of tools with the basic framework of this viewer, and Mastodon is one of them. So you can open 3 windows, which will have the XY, XZ, and YZ options. So for the single nuclei, you have all regions visible, and then the tracking was semi-automated. So you can run the algorithm for, let's say, roughly 10 to 20 frames, and then you curate it. So most of it is actually manual. Only now, very recently in Barbara's lab, what we are trying is to do care with the image restoration and increase the signal, deconvolve, again work more on the signal, and then try segmentation methods, the new deep learning based methods like StarDist. And also people we know are developing more automated tracking methods. So this is something very new. We are just trying it. I don't have full confidence in any one solution yet.
Okay, thank you.
Akansha, I just had a general question. So you work with Drosophila and then Tribolium. Are you aware of many other insect model systems that are being investigated?
So there is a whole zoo that people work on. So the insect community is the Evo-Devo community, and to do this comparative analysis, people are setting up at least one organism from each branch that is possible. So from centipedes, spiders, to Onchopeltis, which is a milkweed bug. So many different, with each branch having some small differences. Most often the big issue has been in labeling. So transgenics and labelings and all of these systems have not worked very well. What people tend to do is RNAi knockdowns, which work quite well, and in situs to see how gene regulatory networks and segmentations have changed. So for my purpose, I am actually very interested in figuring out how this morphogenetic mechanisms evolve. And that's why I am very invested in doing this comparative approach from synthetic to developmental systems. And there, probably with CRISPR knock-ins, again, it would be possible to label. But that's also one of the main reasons I am now learning a lot and establishing a lot spatial transcriptomics, so that we don't always have the need to label, but we can image with a more generic marker and then put an identity to the cells. So if you, like, a simple answer would be, if you were to attend any of these EVO conferences, like there are hundreds of organisms people have set up.
Sure, that's good to know. Is there a general website where we can find all of this? You know, for lazy people like me who wouldn't go and search for every organism.
I think you could just join the mailing list from the Euro EVO. They have a meeting every 2 years, and even if you attend like one of them or see any mailing from them, you would see that.
Yep, that's very helpful for everyone.
The Euro EVO website.
Alex, you had another question?
Yeah, so we, work in developing mouse kidney and, working into the kidney organoid space as well. And one of the challenges in our systems is working a way to, culture the samples that's effective for imaging. And I just wondered if you could comment on how you culture your brain organoids and what the actual setup is on the microscope.
Yeah, so actually I also struggled with this quite a bit in the beginning. So what I did, there was a light sheet course organized in Dresden by Pavel. And at that point, I had gone to almost every booth of every commercial system that was available. And I tried to gauge how hard or easy it is to adapt something. So my personal favorite remains the Zeiss multi-view for doing multi-view imaging, but that wouldn't work for very long periods or organoid systems. So I faced issues with the CO2 stability as well as the temperature stability, and the organoid would be fine for a day, but it's also growing a lot in size, and then you have a capillary based system. So after 24 to 36 hours, this failed. And then from all of these steps, eventually we went to this Viventis Microscopy. This is a new company based in EPFL Lausanne. This is an inverted light sheet, which is specially developed with the idea of organoid or cell culture systems. So it's a very closed system. It can remain very sterile with a little bit of media, and then you can I'm sure for the mouse kind of or kidney kind of application that you're mentioning, I'm pretty confident that it would work with the system where you have a tiny chamber with the media. You can deposit your sample from the top, and you can easily exchange media every 2 days or every day as you want.
Okay. And I thought if I could have one final question. Together with Alberto Rosella Diaz and Toby Merson, we're looking at these kind of, cellular barcoding and lineage tracing options with the view to connect our live imaging back to, organ scale lineage tracing kind of by imaging. How do you plan to map your spatial transcriptomics or your cellular barcodes back to your live images? Do you have the resolution? What kind of approach are you planning in terms of spatial transcriptomics?
Yes. So for the eye tracer part, there I am not developing that. So that was a different story where we had the barcoding system giving us a result about the spatial restrictions, and I confirmed it with the imaging. But from the imaging perspective, I am trying to, so I have these, for example, nuclei labeled lines. On top of this, I'm trying to knock in transcription factors and regional reporters for the brain regions. So this is one way that I have been thinking of approaching this, to track the different lineages in the organoid over time and see how it turns positive for a certain forebrain or a ventral forebrain region. In parallel, what I have been doing is more recently, a lot of whole mount fluorescent in situ or HCR based approach. So I'm able to now label up to 5 genes, and we are trying to make this iterative. So because this microscopy is relatively easy, what we have managed to do is to retrieve the organoids after we image them. And then we fix them, and we try to do this iterative staining on top to figure out how, what are the gene expression patterns across this. And then with analysis side, we are trying to combine this back to the live imaging. And what I'm trying to take advantage is that because no two organoids are similar, and each of them has this very specific topology. So between different lumen, you have like a unique 3D shape. And we are trying to kind of take this from the live imaging side with the tracked lineages and put the gene expression on top in whole mount 3D.
Thanks very much.
All right, Akansha. if there are no more questions, we would like to thank you very much for